Toren, I have a question.
Sure.
Toren, first of all, really a tour de force, unbelievable. Watching, and every question I thought of what I would do next. And oh my God, you did the experiment. So elegant, brilliant. What I wanted to ask was, there's really two parts to the question. The first part is we've often thought in a very naive and simplistic manner about the lysosome acidification. So one thing you talked about brilliantly was, the association of the lipids and the recruitment to ER, et cetera, and turnover. But what happens to the actual acidification machinery, the vascular ATPase, that's important for keeping the lysosome acidic, which was the first part. And the other part was, I was really quite surprised that, and maybe you have looked, that the association or the engulfment of the lysosome repair with the ER wouldn't have triggered an ER stress response, and because of the hydrolytic enzymes. So two parts, two questions. Wonder if you could.
Yeah, both great questions. So for the sake of time, I skipped over. I concentrated, as I mentioned, there are 4 ORP proteins recruited: OSP 9, 10, 11, which I concentrated on. And they exchange PI4P for phosphatidylserine. In the model on the top, there's another OSP protein that OSPP, which exchanges PI4P for cholesterol. And I didn't mention that, but that is actually a parallel pathway for repair. And the reason I bring that up in response to your question is because we do, and this is something that Jay's pursuing, is that cholesterol is very involved in the VATPase, and in crystal structures, it's part of the VATPase and binds directly to the VATPase. And so we do think that part of the pathway is critical for maintaining acidification, but we don't have great data yet to prove that, but I think Jay's pursuing that. So we do think, that part is critical for reinstitution of the acidification. But again, it's still preliminary. Regarding the second question, we haven't actually looked, and it's a great point, to what degree the tethering induces any changes in ER stress responses. And to what degree the lysosomes are damaged, but again, there's no cell death in the paradigm that in wild type cells. There's not enough, the amount of damage is low enough to allow repair if these pathways are evident. If these pathways aren't evident, I don't know if there would be an ER stress response or not. We haven't looked.
Okay, thanks so much. And again, thank you so much for the wonderful presentation.
Oh, thank you for your kind comments. I don't know how to mediate the chat. I don't know if I'm supposed to do it or not, but yeah, please, Avanav, I know you have your hand up.
Yes, that was truly fantastic, Toren. Thanks for sort of laying this out. It makes sense that the lysosome needs mechanisms for repair versus lysophagy and replacement. So how does the cell decide whether this lysosome should be repaired versus this should be replaced?
Yeah, I mean, I think that's, it's unclear, at this point in our minds. I think it's not different than mitophagy. I think, that's the model that I would think of, that, under most conditions, the default mechanism is repair, because, if you can keep the lysosome intact, it's obviously, from a sort of just thermodynamic point of view, it's better to do that than to degrade it and resynthesize. So, I think, I think what the evidence from mitophagy suggests is that there's a gradient of decisions here that, with mild or moderate damage and intact repair machineries, you would go for repair. If you're unable to repair, eventually you would go to lysophagy. But again, lysophagy takes hours. These pathways are invoked within minutes. So that's at least my naive thought at this point about how it happens. I should mention, and I didn't have time to show it, that our pathway and the escort pathway are completely separate. So if we, inactivate escort, it has no effect on our pathway, and vice versa. If we inactivate our pathway, it has no effect on the escort repair pathway. So those two pathways are separate. So we don't really understand, how those two pathways of rapid repair are chosen. They look like they're both initiated simultaneously under most conditions, and both are involved in the overall repair.
So quick follow-up, Toren, is there a role for, ubiquitin conjugation to phospholipids in this decision tree?
Not that we know yet. Not that we know yet. We don't, again, but we're not really looking at that lysophagy stage at this early point. But we haven't directly looked, but we don't think that is playing a role early on, at least.
Lorrie, are you going to read the questions?
Sorry,
Um,
AJ. Sure, I can read the questions. Absolutely. Okay. So, Jamie has a question. Fantastic talk. You think a similar pathway exists in the aged myocardium where autophagy relies on I guess, exophore uptake by macrophages? Is it possible that defects in the pathway in cardiac resident macrophages may underlie the inflammaging, I like that, inflammaging observed in the aged heart?
Yeah, so I think for the most part, I mean, this is again something that Jay's pursuing. For the most part, we with most mitochondrial stresses, we do not see generation of PI4P on the surface of the mitochondria. Jay has found one stress, which again, because it's early on, I don't want to talk about his work, but I think for the most part, the answer is that probably this pathway is restricted to the lysosomes, but there may be occasions where PI4P generation on damaged mitochondria does occur, but again, it's very early on. But I don't think this is the pathway by which most cases of mitochondrial damage is repaired. I think it's unique for lysosomes as far as we can tell.
Great, thank you. Another question. This is anonymous. Great talk. You mentioned that LLOME is a peptide. Is it taken up by cells by endocytosis, and how does it end up in the lysosome?
Yeah, so it's taken up by endocytosis, delivered to the lysosomes, cleaved by cathepsins, and then polymerizes in the lysosomes. And that polymerization process causes holes in the lysosomal membrane.
Great. Another question by Priya Tash. What happens to PI4P labeled ER after it's transferred from the lysosome?
We haven't traced the fate of the PI4P on the ER after it's there. I presume it either it is there's a variety of phosphatases on the ER that remove phosphate groups from PI intermediate. So I assume it's just metabolized, but that's a great question. I don't know the fate, long-term fate of the PI4P on the ER. And that goes back to your question, Lorrie. Are there effects on the ER basically from this pathway? And we really just haven't pursued that with any rigor at this point.
Okay, great, thank you. There are no more questions in the chat.
Okay.
Anybody else want to?
Siba has a question too.
Siba.
Can you hear me? This is Seba.
Yes.
Yes.
So first of all, congratulations for the terrific seminar. I have a few questions. One is going back to the initial demonstration that lysosomal function is impaired during aging. Do you have evidence that the activity of PI4 kinase 2A is progressively reduced during aging?
Yeah, so we don't see any change in the levels of PI, based upon, like RNA-seq data. We don't see changes in the level. We are doing a little bit work in senescence now, senescent cells, and we see that the pathway, that PI4K2 alpha is recruited, but that there are defects downstream, it looks like, in senescent cells in terms of the recruitment of OSPP protein. So there is some defect, we think, in age cells, but we don't really understand at this point where the lesion is. But I don't think it's at the level of 2 alpha or by mRNA or protein, but maybe in its activity or in the downstream factors.
I see. Thank you. And the second question is, we know that the lysosomal function should be highly coordinated with the autophagy function in order to make the system work properly. So my question is, based also on the interesting involvement of the ATG2. So
Yes.
do you have evidence that when PI4 kinase 2A is activated for stimulating lysosomal repair, autophagy is also concomitantly activated?
We don't see that. And in fact, if we completely inactivate autophagy, so if we knock out ATG, any of the essential ATG genes, it really doesn't at all impair this pathway. So we do think of them as being separate, although, as you mentioned, ATG2 is shared between this pathway and autophagy, and it's required for both. But otherwise, we think that of these pathways as being relatively independent of each other. And that was the reviewers, I must say, really wanted us to demonstrate that point. And so we went to great length to show that the role of ATG2 in this pathway is really distinct from its role in autophagy. But so that's at least our belief, that although they share ATG2, these pathways are really separable.
Thank you.
AJ, go ahead.
Yes, sir. Toren, it's fantastic. Really, congratulations. The beautiful PIT pathway discovery. That was beautifully done. My question to you is someone who really is uneducated in this area is, besides genetic mutations that cause lysosomal storage diseases, what other factors can damage lysosomal activity? Specifically, is there any link from cell membrane to lysosome integrity, repair, and activity?
What you mean? Any
From cell membrane, right?
the plasma membrane? You mean?
Plasma membrane, yes.
Any role of the plasma membrane in lysosomal repair? You're saying? No, I'm not sure I completely understand the question, but I think the role of the lysosome and the plasma membrane, the only thing I can think of is that, and this is sort of an area that is pretty uncharacterized, is that the lysosomes can fuse with the plasma membrane and release their contents into the plasma, in the cell, in the body, or into the extracellular space. So lysosomal fusion leaves components of the lysosomal membrane on the plasma membrane. And people think that is a quality control mechanism when the lysosomes are not functioning well and filled with undigested material. One mechanism the cell uses is stimulating lysosomal fusion with the plasma membrane and expelling the contents into the circulation, basically. And so we don't know to what degree this pathway may or may not be involved in that process as well. And that process is poorly understood. What are the regulatory mechanisms that tell a lysosome to fuse with the plasma membrane and deliver its content outside the cell when it no longer can digest them inside the cell?
So would you envision whether a disruption in intercalated disc or desmosome protein to affect integrity of lysosome and leads to lysosomal dysfunction?
Yeah, I don't know the cytoskeleton well enough to predict, what that would be. I think you just have to, look at that directly. I mean, again, there's a number of ways at looking at lysosomal fusion with the plasma membrane, because it leaves LAMP1, which is normally on the lysosomal surface, it leaves it on the plasma membrane. So you can look for the level of LAMP1, which is sort of a legacy of lysosomal fusion, and see whether or not, there is impairment with, if you disrupt plakoglobin or any other of those components or not. I just wouldn't know.
All right, thank you. I think I like to acknowledge Dr. Tagmeyer, who couldn't sign directly, and he signed under my name. The second Dr. Marion really is the god of metabolism on earth, Dr. Tagmeyer, who is signed under my name. I wanted to acknowledge him. He's part of the panelists, and he had issues with signing in. Toren, we're truly grateful to you. This was fantastic. I mean, it's mesmerizing science, and truly congratulations to you for discovering a novel pathway for desmosomal repair and maintenance. And Dr. Lorrie Kirshenbaum, as usual, you are a fantastic colleague, and grateful to you. And I'm grateful to our panelists, Dr. Tagmeyer, Dr. Abhinav Divan, and Dr. Siba Shiareta. I'm most grateful to you all, and thank you, organizers of the Journal of JCA and the publisher. Thank you for organizing this webinar. We are grateful to you all. Again, Toren, fantastic. We're truly honored to have you as a speaker.
Thank you so much. Thank you for your, thanks. Best of.
Thank you.
Bye-bye. Thanks.
Bye-bye.